Hi class, how are you? So today we're going to cover ion and small molecule transport. So oftentimes small molecules like and ions need to sort of get transported across the lipid bilayer, and we're going to look how that's done today. In the first section, we're going to just sort of do small molecule transport and some overview, and then in the second part, we'll start to do ion channels. Okay. So as we sort of talked about when we were covering membranes, they're largely impermeable to ions. Yet cells need to uptake these small molecules, and they also use ion gradients for a variety of purposes, right? So if you think about the proton gradient in between the mitochondrial inner membrane, that's used to generate ATP. You also can sort of think about the sort of moving of an action potential down a nerve cell, right? That is basically the flow of sodium and potassium. And we've seen also in instances with muscle contraction and things like that, and cell signaling, how calcium can play a role in all of these things. All right, and so ultimately it's the transporters that move the molecules across the membranes. And also they can do things besides for energy generation. They can also do signal transduction, and also the pH of compartments is tightly controlled. And we saw this with the lysosome, right, where you had all of the acid hydrolases, and then you had the vacuolar ATPase sort of pumping in protons to lower the pH. And then there's a separate category of transporters called channels, and these just open up and allow sort of ions to flow down their electrochemical gradient. So normally I don't have you guys memorize anything that I show in tables. These are things that you can look up, but you do have to understand some things about this table, okay? And here's the different cations, and here's their intercellular concentration and their extracellular concentration. It's important that you know where the ion concentration is high. So in case with sodium, it's high on the outside. Potassium is high on the inside. Calcium is actually low in the intercellular relative to like the endoplasmic reticulum or the sarcoplasmic reticulum or the extracellular space. And also chloride ion is high outside. Okay, proton you don't have to worry too much about because the differences are somewhat minor. So membranes, even though they're largely impermeable to just a specific class. And these are charged ions. Certain things can actually cross the membrane, and we saw this before with the steroid hormones that can just sort of freely diffuse across the lipid bilayer, and then they can go on and bind to a nuclear hormone receptor. And also you can get small gases like oxygen and CO2 can sort of readily pass the membrane. Small uncharged polar molecules like water and urea, they can travel a little bit, but not really. And then glucose also can sort of cross at some level, but in general, it's the majority of it needs to be brought over through sort of transport mechanisms. So just as a general rule, charged ions are largely impermeable, whereas hydrophobic molecules and gases, they can pass freely. And the way you determine what can go, each of these different molecules have a permeability coefficient. And here's just some permeability coefficients here. So water is way up near the top, so would also steroid hormones, whereas charged ions, they have very low permeability. And if you want to know the sort of the rate of flow just across a lipid bilayer with no transport, what you can do is take the concentration difference between each side of the lipid bilayer and multiply it by the permeability coefficient, and that gives you sort of the rate of flow. So in general, the ones that don't sort of just passively diffuse across the membrane, they're transported by transporters, and the transporters oftentimes can undergo conformational changes. So in this instance, this green is just a protein in the lipid bilayer, and then you have your solute. It could be an ion or glucose or something along those lines, and the transporter will undergo a conformational change which can allow it to pass across the lipid bilayer. And so there's two types of transport that occur. You can have passive transport, which is facilitated, and that uses sort of the existing electrochemical or chemical concentration gradients. And then you can have active transport, which requires some sort of energy like ATP. And we've seen some that use ATP already, and we'll see some more in a little bit here. You also have channel proteins, and these create just an aqueous pore between the two sides of the lipid bilayer. And when they open up, ions can freely flow down their concentration gradient. Okay, so here's passive versus active transport. So let's say you have your molecule to be transported. You can have simple diffusion, where it can just go right across the lipid bilayer. You can have channel mediated, where the solute can just pass directly through following the concentration gradient. If you guys are curious, the concentration gradients shown over here, all of these things are high on the outside and low on the inside. And then this blue molecule will just sort of go down through a transport mediated process. And so this is all considered passive, right? Passive transport. It'll just flow down its concentration gradient. Now, if you want to move something against its concentration gradient, that's going to require energy. Okay, so in this instance, you're using energy to pump this against its concentration gradient. Okay, and so a perfect example of one that uses energy to pump across its concentration gradient is the vacuolar ATPase, right? That's pumping protons into the lysosome, as we saw, to lower the pH. Some other ones that we've seen too were also the calcium transporter that pumps calcium into the endoplasmic reticulum. And we'll look at the calcium transporter in a second, the one that's on the sarcoplasmic reticulum in muscle cells. Okay, so not only is the concentration of the solute important, but also the electrochemical gradients. Okay, so if you have no electrochemical or no membrane potential here, it's going to just sort of follow a certain amount. But the second you add an electrochemical gradient in here and you generate a membrane potential, that's going to help actually drive a positive ion through if it's sort of negative on the inside. Right, the positive ion is going to want to come in to sort of help neutralize the net negative charge that's on the inside. Okay, whereas if you have an electrochemical gradient that's positive on the inside, that actually slows things down even more so than just one with no membrane potential. Okay, so not only is it the chemical concentration, but also the membrane potential can also influence how things cross the membrane and the driving force that's causing them to cross the membrane. Okay, so in certain instances, you're going to have a conformational change. Okay, so in this case, we've got this molecule, this solute, kind of looks like glucose. You can think of it like glucose, and it's going to go in. Okay, along with its gradient. So if this molecule is high on the outside, in state A, it'll just bind, and then it'll undergo a conformational change, and then it can get released on the inside. And so here's sort of the relationship between sort of transport-mediated diffusion versus simple diffusion. So simple diffusion is just going to be a linear line. And as the concentration goes up, the rate of transport will go up as well. However, if you have a transport-mediated process, it's a molecule that's going to be there, so it's going to have a rate of reaction, right? And so, and you can get that just by sort of plotting out how the transport-mediated diffusion occurs, and you can calculate the 1/2 Vmax from it. Okay, and this is going to be dependent on the enzyme concentration, or the Vmax will be dependent on the enzyme concentration. As that goes up, more and more stuff can be transported. And so here's some of the mechanisms of ATP, or mechanisms of active transport. You can have coupled transporters where you can pump things against the electrochemical gradient by sort of letting things passively diffuse through, okay? Or not passively diffuse through, but actively go with their gradient. It doesn't show it here, but the red squares are probably high on the outside, and they want to go in. Okay, and so doing that, it can move a molecule out. You can also use what are called the P-type transporters, these right here, where you have sort of ATP hydrolysis to pump things across. And once again, we sort of saw this with the vacuolar ATPase, which is pumping protons into the lysosome, and also this is what pumps calcium into the endoplasmic reticulum and sarcoplasmic reticulum. Okay, and they're going to use ATP to drive a ion against its electrochemical gradient. Okay, and then you can have light-driven pumps. Okay, and these are sort of, think of them as photosynthesis and things like that, where protons can actually be pumped across based on the absorption of a photon of light. Okay, so here's the transport-mediated movement. You can have what are called uniporters. They just basically will move one single ion. And then you can have sort of symporters, which will move two. Okay, one could be flowing down its electrochemical gradient, whereas the other one is going against its electrochemical gradient, and it can use that. And then you can have an antiporter that's sort of transporting one out and one in. Okay, and these are sort of coupled transporters, where they're sort of using the natural electrochemical gradient to drive things against their electrochemical gradient. And you can sort of see that with the sodium-dependent glucose transporter. Okay, so this is going to be sort of one of these symporters right here, where you have sodium concentration high outside the cell, right? We sort of covered that when we looked at that table. Sodium is high outside, and the cell wants to take up glucose. Okay, so glucose will normally be higher inside the cell. Okay, so you can sort of think of this almost as the sort of the intestinal epithelium, where you want to be taking up glucose in food sources. Okay, so they're going to use the sodium to actually move glucose against its concentration gradient here. Okay, and it's going to use the sodium that's outside. And then it just undergoes a conformational change, and the glucose can get released in. Okay, so the sodium's going to flow naturally down its electrochemical gradient, whereas the glucose is going against its gradient. So in this instance, where you have this sort of symporter here, it's using the sodium to drive the movement of glucose. And you can see that too with lactose permease, LAC-Y. This is actually part of the lac operon in E. coli. And so the way it works is it uses the proton gradient to uptake lactose. Okay, and the proton ions sort of work as the symporter. The proton ions work with lac permease, which is a symporter, to move lactose across the membrane. And here's sort of the mechanism by which it works, because you can sort of think this is moving back and forth, and it should be able to just open and close and move back and forth, but it doesn't due to sort of the electrochemical properties of the actual enzyme, the lac permease. And so here's a sort of a good example here. Normally it's in its open state, waiting for some lactose, and a proton comes in and will sort of neutralize the acidic residue in the glutamic acid here. Okay, so it's binding to the COO that's sort of present on the R group in glutamic acid. Okay, and so when that's bound there, the lactose can come in, and there's a sort of a forbidden state here that holds it in an open conformation. It can't go backwards because of these two positive charges are sort of preventing it from sort of closing on itself. But once lactose is present there, it actually can, and then all of a sudden you have the arginine here on 144, which has a free amine and a positive charge, and it can sort of neutralize the glutamic acid, the proton, and the lactose get released. Lactose first, and then the proton, and then it can go back to its sort of resting state here where it's ready to go for another round again. And this happens in our cells too, and we sort of already covered the sodium-dependent glucose transporter. So sodium is high outside. This is an intestinal epithelium. And so here you have high sodium outside, and you want to uptake the glucose, and so you're going to have this symporter helping to drive glucose, which is then high inside the cell, and then you can have a glucose uniporter, which just allows glucose to sort of pass into the extracellular space into the body. So this is just one of the ways that we sort of take up foodstuff from our intestinal lumen and across the cells. And meanwhile, sodium is actually being pumped out by the sodium-potassium pump down here, so that the flow of sodium can drive the flow of glucose. So low glucose outside, high glucose inside, and then it'll just flow down its concentration gradient. Whereas sodium is high outside, and it's going to flow in, and then you have the sodium-potassium pump pumping the sodium back out. And as we look at this figure, I also want you guys to sort of think back on how we were talking about protein sorting. And so the cell does need to know how to get the transporters to the right location, right? This transporter needs to be on the apical surface, and this transporter needs to be on the basolateral surface, right? So we were talking how certain proteins are destined to certain regions on the cell when we were talking about transport, and this is just a good example of two that need to be separated, because if they weren't transported to the right location, this process couldn't occur. Okay, so here's some of the active transporters, the ones that use ATP. And so you have the P-type. This is the vacuolar ATPase that pumps protons into the lysosome, and also the calcium pump that pumps it into the endoplasmic reticulum or into the sarcoplasmic reticulum. And they're going to use ATP hydrolysis to pump the ions across and against their electrochemical gradient. You also have the F-type, and this is basically the pump that's used to generate ATP, and we'll see this when we cover oxidative phosphorylation. So in the inner mitochondrial membrane, the level of protons are quite high, and they will flow down, and as they do it, you have a little rotator there that's going to convert ADP, an inorganic phosphate, to ATP. These can also go backwards, so they're reversible, where they can use ATP and they pump protons out against their electrochemical gradient. But the way it's shown here, this is basically the F-type proton transporter that's used to generate ATP in the cell. And then you have your ABC transporters, which we've seen more than once now, and these are going to use ATP hydrolysis to change conformation and open up, and in doing so they can transport small molecules. Okay, so like I said, we'll look at these a lot closer or this particular enzyme, when we look at oxidative phosphorylation. They have the crystal structure of it. It's pretty wild. So we'll look at that later on. But in the case with the P-type here, here's the calcium pump that's actually in the sarcoplasmic reticulum. Okay, so this protein actually makes up 90% of the protein in the sarcoplasmic reticulum membrane. And here's how it pumps calcium into the lumen of the sarcoplasmic reticulum. Okay, so if you can see, it just undergoes that conformational change, and there's a phosphorylation event that occurs. Okay, so in its sort of open configuration, calcium will go into the calcium binding site, and then you have a nucleotide binding domain, and that's going to end up leading to phosphorylation of aspartic acid in the phosphorylation domain. That's going to cause a conformational change. No more calcium can get in, but it'll open down at the bottom and 2 calciums can get out. So this is what's constantly keeping the calcium low outside the sarcoplasmic reticulum, right? And so this was sort of the enzyme I was thinking when I said if you added an inhibitor of calcium uptake in the question on the last exam, this would be the enzyme that was getting inhibited, okay? And so the calcium would stay high outside if it couldn't bring it in, because it would just flow out. Okay, you also have the sodium-potassium pump, and I'm sure you guys have all seen this at some point in time in many other classes. But once again, sodium is high outside, potassium is high inside, and you have the sodium-potassium pump that actually pumps it, so it's doing that. So it can move 3 sodium ions out and takes 2 potassium ions in for basically using 1 equivalent of ATP hydrolysis. And here's sort of the mechanism by which it works. So it's normally, you know, if you start here, sodium will flow in and potassium flows out, and then the enzyme will bind ATP and undergo a conformational change. You have your 3 sodium ions, and then it gets phosphorylated, and the ATP is hydrolyzed to ADP. The ADP is released, and that opens up so the 3 sodiums can go out, and now 2 potassium ions will go in. And then when the protein gets dephosphorylated here, it's got the potassium in, it'll undergo another conformational change, and then it just opens up and the potassium can flow in and the sodium flows out. Okay, and this is basically used to establish the sodium potassium gradient, where sodium is high outside the cell and potassium is high inside the cell. And this is basically present on every single cell in our body. And it's also very important in nerve cells to maintain the concentration gradient so you can propagate an action potential down a nerve cell. And we'll see that when we cover the ions channels in the next part of this lecture today. The other thing that the sodium-potassium pump does, it actually controls cellular osmolarity, right? So here's just a red blood cell, and this is normal, and it's sort of considered isotonic, and so the potassium and sodium levels are just perfect. But if it gets too hypertonic and there's not enough water, it looks like this. If it's hypotonic and too much water is entering into the cell, it gets sort of swollen, and if it gets too big and very hypotonic, it can actually lyse. Okay. So the sodium-potassium pump has a direct role in controlling the osmolarity of a cell. So it's important in every single cell in our body. Here's the ABC transporters. So once again, we've seen this exact figure before, where in bacteria it'll be involved in small molecule uptake. So the small molecule will come in to the ABC transporter, and then two equivalents of ATP are bound, and that causes a conformational change, and the solute molecule starts to travel in, and then it hydrolyzes the ATP, and it goes back to its resting conformation, and the molecule is then completely across the membrane. In eukaryotic ABC transporters, they're oftentimes involved in pumping things out. So here you have a small molecule in the cytosol. It'll flow in, and then the ABC transporter will bind ATP, and it changes conformation, and then it can sort of move it into this sort of vestibule area here, and then it hydrolyzes it and goes back to its resting conformation, and the molecule can just sort of flow out. And these are actually a pretty large family of transporters. 5% of the E. coli genome actually encodes for these ABC transporters. And here's sort of the E. coli double membrane. So the ABC transporters are actually in the inner lipid bilayer, and then in bacteria you have this sort of outer membrane that has lipopolysaccharides and porins, so things can freely diffuse across the porins. Then you have in this periplasmic space oftentimes a bunch of carrier molecules, and then the ABC transporters are in the inner lipid bilayer, the inner membrane. And here's sort of how it works. So once again, small molecules can freely diffuse through the porins, and then they have a substrate binding protein in the periplasmic space that will sort of catch the molecule, bring it to an ABC transporter, and then using ATP hydrolysis ATP binding and hydrolysis, it changes conformation and releases it to the inside of the cell. Okay, so that's it. In the next part of the lecture, I will go over ion channels. Okay, thanks.